Thank you very much for your attendance. So we're continuing the distinguished lectures at the Josep Carreras Institute, and today we have the pleasure of having with us Dr. Dominique Bonnet. So this is the kind of the John Dick week. We were joking because at one point she was trained also in that lab. Dr. Dominique Bonnet, she obtained her PhD degree at the University of Paris. And then she, as mentioned, she joined Professor John Dick Lab in Toronto, in Canada, for the postdoctoral training. After 4 years later, she accepted a position as group leader at the Coriell Institute for Medical Research in New Jersey and became an assistant professor in the University of New Jersey. In 2001, she went back to Europe, not continental Europe, but Europe, at Cancer Research UK in London, as a group leader. In August 2012, she became a professor at the University College of London and a senior lecturer at the Institute of Child Health. In 2016, she moved her group to the new Francis Crick Institute, that is a top-notch place for research in Europe, and her group is investigating the molecular programs that regulate normal blood stem cells and the oncogenic events related to this process, and also now a focus in the microenvironment in these models. And she has recently developed a humanized niche model to further study the interaction of hematopoietic stem cells and the bone marrow niche. And the title of her talk relates to this, is the bone marrow niche, a supportive ecosystem for haematopoiesis and malignant cell growth.
Thank you very much. Thanks for the introduction and thanks for inviting me today. So as Manuel mentioned, I'm going to do a lot of PDX talks. So you have heard John Dick, so you should be familiar with that. The only differences, I suppose, is today I'm going to focus much more on the interaction between MDS, AML, and the microenvironment. So the talk will be very much focused on the complexity of the bone marrow microenvironment that you can see here. And when we started a few years back this kind of project, the only thing we know is that leukemia in general, whether it's MDS or AML, even more for AML, is that they are very difficult to keep alive without a stroma population. So we know that the AML needed some sort of support from the microenvironment. We also did some work on hypoxia, which is also known to be present in the bone marrow, and we know that both normal and leukemic stem cells are really maintained by the hypoxia and mostly if2 on these. So we thought that there was not much work about it at that stage, and we wanted to know how leukemia, whether first leukemia impede the environment, what is changing, and can we learn about that and maybe impede indirectly the leukemia growth by these. So the first vignette that I'm going to show you was started actually some imaging that we developed in the lab in order to see where these leukemic cells go. We inject them into these immunodeficient mice. So it's patient samples that we inject, but we didn't really know, except looking 12 weeks later, whether the leukemia engraft or not. We didn't know exactly what happens in the mice and where they were and how we can look at these. So, we developed this intravital imaging. Is a relatively non-invasive way, as we only image the calvaria of the mice. So we do an incision of the skin, but then after we don't go inside the bones, and we use 2-photon microscopy in order to go inside the bones and look at it. And this is just to show you that if you inject a GFP leukemia, you can see the progression of the leukemia engraftment and evasion into the bone marrow. And when we were doing that, one of my students at that time said that there was the leukemia really liked to go on top of the vessel. And then he did some imaging and he left, he did this PhD and left. And I had look at these pictures, and I was still very interested by the intriguing things that in a control, you see the very nice structure of the vessel, but when you inject either a cell line or a primary patient samples, when you look at the vessel structure, all the blood contrasting agent seems to go completely outside the vessel. And so it took quite a few years back after from another person, a postdoc, Diana Passaro, to really investigate that a little bit more deeply and quantify these permeability or leakiness that we saw originally a few years back. And you can see indeed that is very specific to primary AML patient, as if you inject normal stem cells from umbilical cord blood, you don't see these appearing. So she did some transcriptomics analysis in order to understand the mechanism of action of what was happening in the endothelial cells from these mice. And what we find is that there was clearly an angiogenesis coming up, there was a vascular development, but also there was a pathway that she was interested in, which is an increase in eNOS or NOS nitric oxide synthase that was very present. And I wanted actually just to further discuss that. I think it's important to see is that these different patient samples that we had different genetic makeup. So it's not necessarily based on a specific mutation, but it was actually the group very closely together, indicating that it's quite a common thing to have. So I'm not going to go too much in detail. I'm going to show you the graphical abstract of the paper that we published on that, just because it's been already a few years back, and this is already, you can get more definition on this if you want. So what we find essentially is that with the leukemia, we have an increase in the signal of endothelial cells of these NOS, especially NOS3 and NOx, which essentially secreted nitric oxide, and nitric oxide has been shown actually even in solid tumor before to be involved in leakiness of the vessel. And what we also find is that with that, there's an increase in hypoxia, even more drastically in acute myeloid leukemia. So what we also find is that if we treat the mice with nitric oxide inhibitor, we actually decrease the permeability of the vessel, and we can actually cure more specific the leukemia by the normal chemotherapy. So just impeding in the leakiness could actually prevent or actually ameliorate the chemotherapy treatment that these mice receive, which are actually what the patient receive. So this is one of the examples of the crosstalk that I mentioned between AML and the endothelial cells. But of course, I mentioned the bone marrow is a very complex microenvironment. So we end up also looking at over potential population of cells that may be important, reported to be probably important for the maintenance of both normal and leukemia. And these are essentially all these markers who we look at endothelial cell, mesenchymal cells, perivascular, progenitor, osteoprogenitor, and osteoblastic. And this was done by actually crossing all these NAG mice with reporter mice that we can easily sort the population after injecting the leukemia. And we did quite a lot of RNA transcriptomics in order to look at these in different AML patient samples. And we first find different things, which is that different reporters that have been shown to really characterize specifically subpopulation of mesenchymal stromal cells actually were quite overlapping in between each others. But what was interesting when we merged all these data, we saw actually that not only, so of course, leukemia impede a number of different pathways in all these different subpopulation of cells, but also what was probably less known is that there's also a very big crosstalk between cells of the microenvironment, especially the endothelial cells talk to the nestin positive cells, for example. And we have actually now a number of targets that we are further investigating. Unfortunately, I don't have much data to produce you, but we actually, if you are interested in specific genes, you can then interrogate these large homing as its repository in this paper, and you can enter the gene of interest, and you will have some sort of data coming up whether or not we pick up these either upregulation or downregulation, and in specific subpopulation of stroma cells. So this is something that we hope will be not only important for us to follow up on all these different targets, but also for the community to work with us on this. And as Manuel mentioned, one of the things that we were still, and we always have criticism about looking at AML in context and the microenvironment in the context of a PDX, is that we inject human cells, but we are looking at mouse stroma cells. So in order to try to move away from the bone marrow niche, mouse bone marrow niche, we try to engineer a humanized niche that will recapitulate much more human bone marrow microenvironment. And this is with the work from Ander Barat-Teji, a postdoc in my lab at that time, now group leader in Bilbao. And what he did, essentially, try different type of scaffold. And one we are using still even nowadays is a scaffold, is a sponge of collagen that we use as a scaffold to inject human mesenchymal stroma cells. Now, actually, I'm not going to show you much on that, but we also inject human endothelial progenitor cells into this scaffold, and we inject any type of cells. So far, we have looked at normal stem cells, but also AML, and what I'm going to show you more, MDS. And with these scaffold, we find a good level of engraftment. The advantage of this also is that after 14 weeks, usually we wait, you can dissociate these scaffold with a collagen, and you can do a fax analysis and take the stroma cells or the mouse cells or the human cells and do single cell or whatever you want with it. And this is interesting because we believe it will produce a mini niche, and all the constituent of the niche, we see adipocyte, we see osteoprogenitor in the niche represented here in this, and we get very good level of engraftment if we inject normal bone marrow stem cells. And we are, of course, further evaluated this model as we speak, but I think it still works very well already in our hands. What we wanted to do, and I'm going to show you specifically these today, is that we were very interested in the progression of AML, from MDS to AML. And we know that in myelodysplastic syndrome, 20 to 25% of the patient will indeed transform. But MDS has been very hard to develop a model using PDX in the past, and we published a paper which essentially show very little engraftment ability of this patient. As you see, it was in between 0.1 to probably 5% of the engraftment after injected quite a lot of cells. It was actually shown by Andreas Trump's group at the time where we were doing this work, that by co-injected human mesenchymal stroma cell from the patient, you can actually increase the engraftment of these cells. Nevertheless, in our hands, whether it was normal MSC or patient MSC, co-injected intra-bone in the mice, we didn't see much improvement of that. But now, as we have this scaffold, we thought that now we could really understand the crosstalk, because in this case, even if you inject intra-bone the mesenchymal stroma cells, after 1 week, we couldn't detect these mesenchymal stroma cells, so they were not long-term engrafted into our mice. So now in this scaffold, what he did is quite intensive work, because you have to get the patients, you have to expand the MSC from the patients, keep some CD34 positive or mononuclear cell from the patients. Then when you have enough MSC, you essentially did do all these co-injection into the scaffold, implant into the mice, and look at the engraftment. But then you see that the level of engraftment, whether you are different type of MDS from low risk to high risk, you see that we got much higher engraftment, sometimes around even 80%. Still not as good as AML, but still much, much better. And what we find is that we look at different mouse model that we have, so NAG and NAGS, which is humanized for stem cell factor, AL3 and GM-CSF. We have quite similar level of engraftment, so we can use whichever we have. But what was interesting also is that the engraftment is quite different compared to the ML, and it's reproducing what is in the patients with a more myeloid development, specifically in the more high-risk MDS compared to the WAS, for example. So we recapitulate the type of engraftment that we see in patients. The question was, could we also have the mutation burden, and whether or not we indeed see the same mutation pre and post. And this is one example. I think I will show you these 3 examples, but yes, indeed, this is the patient that we injected, and this is the cells out of the scaffold 14 weeks ago. So we indeed seems to promote the engraftment of the MDS clones in our model. What was also interesting is, because these mice were highly engrafted, as I mentioned, in the scaffold, we wanted to see whether we can see something in the bone marrow of these mice, and you can see 0 engraftment for most of the time, except these two small dots here. So we wanted to understand what was happening. Is it because MDS do not go outside, they don't migrate, or is it that they cannot colonize the bone marrow of these mice? One possibility is that the fact that we are using NAG or NAGS, and in order to get good level of engraftment, usually you have to sublethally irradiate the mice. And we didn't want, of course, to irradiate our scaffold, because the idea is to look at the stroma environment. So we make the trick of moving to this model that we have in the lab, which is an NAGS kit. It's a kit mutations in the mice, which allowed actually to engraft either cord blood, AML, or other disease without conditioning the mice. So we use these mice as a recipient to our scaffold, and you can see if you inject healthy bone marrow in the scaffold and just look a few weeks later at the engraftment in the scaffold, but also in the bone marrow of these mice or the spleen, you have indeed dissemination of the normal stem cells coming up. So with these, could we see indeed the same thing with MDS patients? So this is a summary of a few adult bone marrow that we have engrafted. This is the level that is in the scaffold, and this is the secondary mouse bone marrow and the spleen. And you can see clearly that is quite regular what we see. So now we do the same, but instead of putting adult healthy bone marrow, we put MDS stem cells or mononuclear cell T cell depleted into the scaffold. We see good level of engraftment in the scaffold, but nothing in the mouse bone marrow. They're still empty. So one possibility is that they don't colonize the bone marrow, or they don't migrate. So in order to eliminate the possibility that there's a problem of migration of the MDS, what we did is implant 2 different scaffolds. One scaffold where we seed the MDS stem cells with the MSC, of course, but one where we only seed the mesenchymal stroma cells, and wanted to evaluate whether these cells, that is here and planted here will migrate to the empty scaffold, as we say. And indeed, it's what happens. You can see here that there's a primary scaffold that was seeded with the mesenchymal, the MDS stem cells is engrafted, but also we have populated engraftment of these MDS stem cells in the adjacent scaffold. And this is true only if we use primary MDS patient or healthy MSC from the healthy donors, but not if we put in the scaffold mouse stroma cells. So these, I wanted to essentially summarize this work. These also have been published quite recently, end of last year, showing that indeed the MDS hematopoietic stem cells are highly dependent on the humanized niche. They seems to actually quite actually even engraft better if you use mesenchymal stromal cells instead of their own patient's MSC. And this is something we would like to investigate further. And they migrate as long as there's a niche that is available for them to do that. So how can we understand a little bit better these interaction? So I'm going to show you something that we are just in a process of doing at the present time, and something quite simple is a co-culture of healthy donor or MDS stem cells in contact with stromal cells or mesenchymal stromal cells, either healthy or MDS derived. We co-culture these for 5 days, either in contact or we do transwell, and we did some RNA-seq on that on the stroma cells. And what I'm going to present you is only the stroma cells. But just first to show you that in just looking at the expansion of the cells, whether they are healthy or expansion or MDS stem cells, you see that both like the stroma support for the expansion, and this is either from a MDS MSC donor or healthy donor. As I mentioned, there's a good expansion, and this is also true, maybe a bit less if you use healthy donor MSC, healthy donor. So what is the transcriptomic changes that we see in these mesenchymal stromal cells? And surprisingly, what we find, and maybe not surprisingly based on the in vivo data that I show you, is that you can see that the healthy, so healthy MSC with healthy hematopoietic stem cells, different donors are clustering together, but the MDS stem cells with MDS MSC are in this corner, and quite striking, the MDS stem cells that have been cultured only for 5 days with healthy MSC, the MSC have actually seems to have unpicked a disease transcriptomic in a few days only. So we are looking into these in more details. This is just another example to show you, in another way to show you that the MSC from MDS or from healthy co-culture with MDS stem cells have very similar transcriptomics only after 5 days. So we wanted to further investigate that. We have used these Antireactant, that this software that is called NicheNet, that allowed us to essentially look at ligand receptor interactions. And so far, we could also give you also a score of the interactions. So all these very thick line between these different compartments indicate that there's probably a crosstalk between HSC in the left and MSC in the right. And we are actually at the present time looking to validate some of these targets or these communication between the two, and seeing whether or not that indeed help us to see the minimum crosstalk between the two that are important for the MDS stem cells to proliferate in our system. So as I mentioned, MDS cells initiate and maintain a malignant transcriptomics in the bone marrow of the MSC, even if there's MSC normal. It seems to be very much of a rewiring of the intrinsic core of the MSC, the bone marrow MSC. In direct contact, this doesn't appear to be very much happening in transwell. So we have this antireactant ligand receptor now that we are looking at, and we are started a large-scale screening of these in order to see whether these impede or give us a clue of the biology of the MDS disease. So another example of the crosstalk, and this is something that we have worked in my lab for some times, but we have struggled to go mechanistic with these, is the fact that in AML this time, go back to the AML, is that AML actually patients quite often die not only for just their presence of leukemic blast, but because they have anemia, hemorrhage, and they have a severe infection. So what we call bone marrow failure, even though it's not really the term, but really they do indeed have a lot of problems, and they die mostly from infection on over non-leukemia specifically. And this is actually this type of anemia, hemorrhage, and infection is actually not dependent of the bone marrow infiltration. So you could have a very bad, all of this syndrome could happen with even less than or just 20% of blast in the bone marrow. So it seems that we still didn't know much about it. So one of the paper that we published actually 10 years back, almost now, is to understand what was happening in the normal residual stem cells of these patients. So we took advantage of AML that have a nucleophosphine mutation, as we saw that we published that their leukemic stem cells are essentially in the CD34 negative compartment. So we can essentially quite easily know that if we have CD34 positive, and we confirm that, of course, genetically, that these are a part of the normal stem cell, residual stem cells. And when we look at the bone marrow aspiration of this patient, what we can see is that there is actually a quite normal level of stem cells compared to healthy, which is in white here, but there's a decrease in their progenitor. And when we go a little deeper into the analysis of the stem cells with what we call allogeneic phenotype, you can see that indeed this is actually even more pronounced, that the more primitive population of cells, residual normal cells present in the bone marrow of this patient at diagnosis seems to be actually even more enriched than in normal. So what is happening and how these cells are there, but they don't seem to produce progeny. So one of my PhD students developed questions around that and wanted to know whether this is due to the fact that AML have secrete or in contact with normal hematopoietic stem cells do something negative or inhibit these cells directly or by secretome of something, or whether the niche again is implicated in this crosstalk. And so for that, he did a very simple experiments where he either co-culture CD34 positive cord blood and with AML, and before culturing these, he used a cell tracer in the CD34 positive. So if the cells divide, the dye will be diluted over time. And this was done with or without the mesenchymal stromal cells. And you can see here in both these diagram is that if you take these patient samples, actually it's a cell line here, but we did it with patient the same thing. You have the CD34 positive that started to essentially proliferate, but with co-culture with AML only, you see that you reduce the proliferation. And this is even more striking if you have the MSC in co-culture. This is the absolute trace of the violet that was present in the CD34 positive. So with MSC and AML, you essentially that you skip the cells in a more quiescent stage. So because we know by doing the transwell that it was something secreted, but what was important is that you needed AML in direct contact with the stroma, and then something is being secreted here that impede the normal hematopoietic stem cells proliferations. So we look at transcriptomic, actually microarray analysis some years back that we still had in the drawer, and look at either stroma cell with AML cell line or stroma cell with AML patient sample that we have. And because it was happening in both cases, we were looking at the shared genes that were there. And more specifically, we were looking at something that was secreted. So we had actually end up with a relatively limiting number of potential candidates. And one of these was the one that we follow up because nothing was known into these genes. It's called Stenocalcin 1, and Stenocalcin 1 has been shown actually as a poor prognostic in many solid cancers. But in AML, nothing was known about these genes. And the advantage of the Stenocalcin also is that you have recombinant Stenocalcin that you can buy. And so we did the same assay working with CD34 positive from covered healthy MSC, so nothing is pathologic here, but we added a recombinant Stenocalcin and asked the same thing, will the addition of Stenocalcin impede the proliferation and restrain the differentiation of our CD34 positive? And it is indeed the case. We have more HSC-like cells, less progenitor by adding recombinant Stenocalcin. We further confirm the functionality of these cells by doing a long-term culture initiating cells. And this is the frequency. You indeed preserve the more primitive fraction, and if you do a transcriptome of these cells, you see that they have a higher stem cell signature in the Stenocalcin treated part. And again, the importance is to go back to the clinic and see whether Stenocalcin could be indeed present either in the bone marrow. So we did some bone marrow fluid of patients, either a diagnosis or a remission, and you see that there's increase in Stenocalcin in these patients. And even if you take the peripheral blood, which is easier to obtain from the patients or healthy individual, you see that indeed Stenocalcin is higher in the blood, in the plasma of these patients. We could also correlate the level of Stenocalcin with the thrombocytopenia present in these patients, indicating that possibly it's one of the players that could be involved into these problems that we see in patient samples. So I'm summarizing this work also. Here we have done a little bit more on the molecular characterization of the mechanism. What we find is that AML in contact with mesenchymal stromal cells, one of our favorite genes came up, which is if one is being stabilized by the co-culture, even in normoxia, and one of the target of if is a Stenocalcin. So Stenocalcin being secreted. What we don't know is how Stenocalcin is stopping the cell cycle of the HSC, and something that we are still continuing in studying in more details. But what we believe is these are at least responsible in part for what we see in patient samples. So summarizing a little bit so far what I show you, and just a cartoon, but it's complex and we are not there yet, but and I just show you some of the vignette of what we know so far. There's some more literature that are present here, but so far we know a little bit of the MSC and the MDS and AML. What we know also is the data that I presented you so far, that MSC could also impede normal hematopoietic stem cells. But as you see here, there's different stroma cells to be looked at, and we are just at the very start of these. So these indicate that if you are interested in the field, you can jump because there's enough work for everybody to understand what is going on. So now in the few minutes that I left, I'm going to present something a bit different. I mean, still related to the haematopoiesis and leukemia. But as I mentioned in my lab, we were interested in MDS and AML and trying to understand actually the why certain patients are not always transformed into AML, but also during these 7-8 years now, there's this notion of prelocymic events that occur. So we now know that, you know, a number of us over than 50 may actually already have this prelocymic mutation in our blood. And we know that these patients that have prelocymic mutation have a higher risk to develop any of these disease, but also potentially also now is clear that they also have potentially associated other disease like cardiovascular disease, inflammatory and pulmonary disease, and severe infections. And I think the list start to increase almost every week, so it's kind of difficult to follow up. But so what we could do with that and where we could be interesting in understanding so far, what has been shown in the mouse model mostly is that certain environment stress could help the expansion of these prelocymic events and help the evolution of the disease. But what stress is specifically and how this could affect the clonal expansion of this mutant clone is not yet completely clear. And also what is even less clear is that these mutant HSC are producing progeny, and very little, except a bit on monocyte macrophage, very little is known about whether these innate immune cells, more specifically in our case, we are interested in innate immune cells, whether these progeny that have the mutation are actually helping to the evolution and the expansion of the clone. And that's something that we have started to look into. So the approach is always the same thing, except now we're using either normal adult bone marrow umbilical cord blood. We added these prelocymic mutation by CRISPRizing these cells. Here today I'm going to talk only on TET2, and then injected as normal in our NHG mice and looking at the human hematopoiesis system in different. And what we wanted to study is how these mutant clone compete with the normal non-mutant HSC, and whether a certain external factor indeed affect this competition, and more specifically also how these mutations affect the progeny of these cells. So we started ex vivo, and this is something that was quite easy. LTSIC, you do a 50-50 ratio between mutated C34 and wild type. And if you do nothing to the culture, you see in white here, you have essentially 50-50 of the clones that expand, progeny of the clone that expand. If you now treat these cells chronically with LPS, you see an increase in the TET2 mutant clone quite dramatically. So we wanted to reproduce that in vivo to see whether it was the same. And here we took the trick of mixing HLA A2 positive versus A2 negative and reversing it. So we didn't have any bias of that, and injected 50-50 into our mice, a mixture of that. If you do nothing to the mice and look at 14 weeks later at the human CD45 positive, you essentially see that actually the TET2 mutant clone is disadvantaged in the growth. There's much more white type cells there. But if you treat your mice with LPS, mimicking what we've seen here, you essentially change the ratio in favor of the TET2 mutations. So what was happening and what is happening at the hematopoietic stem cell level, for that we did some RNA-seq on highly purified hematopoietic stem cell coming out of the mice in order to look at the transcriptomics analysis of these. And I'm just showing you a few slides here, but what we find, which was what almost we expected, if you look at the normal cells that have been in contact with LPS compared to the control, you have inflammation coming up with interferon gamma and interferon alpha response, but also a lot of interferon stimulating genes that are upregulated. All these in red are upregulated. Now, if you do the same, but compare the TET2 LPS versus the white type LPS stem cells, then the most top dysregulated gene between the two are actually the same genes. So that means that the TET2 mutant are actually refractory to the LPS as mediated transcriptomal change. They don't respond to these inflammatory signals. And we believe this is helping these cells to slowly expand over time. Of course, more data, more study need to be done in order to evaluate that in more detail. But what I also wanted to show you is what happened to the progeny. So this time we didn't inject in competition to make the life easier for us. We inject either the white type or the TET2 from the same cord blood, so that's the same stem cells. And you can see that when you sacrifice the mice, usually you get biased for B cells, and if you get the TET2 mutant, there's more myeloid cells. In order to go a bit deeper into the phenotype of the mature cells, we essentially develop a panel of cytoff antibody in order to further dissect this different population of cells that were present. And we find out that actually there was 20 different subset of subclass of progeny that we can find transcriptomically by cytoff, and the one that are really changing between the control and the TET2, and you see some of these appearing here, actually are mostly focused on granulopoiesis. So there was a lot of different subtype of subset of neutrophil that was changing. So we ask bioinformatically to work cluster these CD66B granulocyte and further evaluate these. In normal cells, what we find is that we have 5 cluster of neutrophil, and these come from the more progenitor type that express 117 to the more differentiated one. And when we do the pseudotime, indeed, these are the N1 are the more primitive, and the N5 and 4 are the more differentiated. Interesting to mention is that these type of analysis and clustering are very similar to what you can get from adult human bone marrow. So we recapitulate very much the neutropoiesis coming up in the mice. So now what is the difference with the TET2 mutant clones? And you see that the proportion is quite different. We have a loss or decrease in the N1, the more primitive, and an increase in mature cells. And this was confirmed by the normal FACS analysis, mature neutrophil increase, immature neutrophil decrease. And if you look just at forward and side scatter, the TET2 also appear to have low granule complexity. So based on the fact that I mentioned that we did a point mutation in the TET2, we are expecting to have a null mutations. So in order to prove that this was happening, we did some DNA methylation and ATAC-seq. And this is just to show that the neutrophil from the TET2 mutant are indeed more hypermethylated, and this is distributed across the genome. And by ATAC-seq, we can confirm that by showing a more condensed chromatin and less peak in the TET2 mutant neutrophil. So transcriptomically, what are the big change? We find actually that some of the change are related to this granule that I mentioned. There's actually more upregulation of primary granule, downregulation of some, and actually some of these could be actually also involved in nitosis, which will be interesting later on. But if we compare these RNA-seq that we have to already published dataset from human bone marrow neutrophil, our control MSC neutrophil looks very close to a mature neutrophil, and the TET2 seems to be more like a pre-neutrophil. So it's quite strange somehow, because at the surface I mentioned it looks like you have more marker of maturity, but at the transcriptome level it seems to be more immature. So what is happening also is we have a decrease in phagocytic genes and pathogen recognition. All this is downregulated in most of the TET2 neutrophil. So the question was, is this functionally relevant? And indeed, if you take beads of zimazine and you put your neutrophil, either control or TET2, you see that these beads are phagocytized by the neutrophil normal, but not the TET2, and the same with Candida albicans here. So indicating that the change that we see in transcriptomic are indeed having a functional effect. And neutrophil is also known for their nitosis. So we look into net, and you can in vitro reproduce the net by using different components. We use PMA, and you can follow up the nitosis going on. And the green signal correspond to the DNA trap that is being released by the neutrophil, and how you look at that. And you can look at the timing of this, and you can essentially what we see most strikingly in this net coming from TET2 is that they are more condensed and shorter, smaller, sorry. And these if the video show off, you will see that in action. Not sure. Maybe not. So this is the TET2 mutant, and this is the normal. See that the net are more splashy, more diffuse compared to the one in the neutrophil one that I show you here. You see much more condensed green cells. So the nitosis actually is a very important process for fungal infections. So we wanted to know whether these, as in the fact they are smaller, they are more condensed, is this really important? So we use these hyphae fungus, and they grow happily without neutrophil. If you use these human neutrophil, the control actually control the infections, but the net coming from the mutant neutrophil are less efficient at doing that. When the net is being done, they need to be degraded, and this is important for that the clearance of the net is also important to stop the inflammation. And this is done actually usually by the nucleus that are present in our plasma. So this is what we see in the control. You take the nitosis is induced. Without plasma, they perdure quite a lot. And if you use different healthy donor plasma, you essentially eliminate this net quite rapidly. And then now if you compare the net from control versus the TET2, they actually are less clear easily. And this, the net being more prolonging and less clear could have a potentially influence, as I mentioned, as inducing chronic inflammation and possibly also aggregation of platelets and possibly thrombosis. So from this work, our working model at the present time is the fact that inflammatory stress, at least for the TET2 mutation, seems actually to increase the expansion of the mutant clones. These mutant clones produce aberrant neutrophil, which is what I focus today. And these are the consequence of that with impaired phagocytosis, aberrant nitosis. So we are in discussion with the clinician at UCH, actually, which are looking into whether TET2 mutant patients have actually more fungal infection. And this is something we are trying to get the data out of it. But these, clearly these NET problems and the neutrophil problem may have also over direct effect on other problems associated with prelocymic mutation, such as probably thrombocytopenia or thrombosis possibly, but also inflammatory pulmonary disease, which could be involved the NET. And I just wanted to thank people in my lab. I mentioned the work of a number of them, so I'm not going to details. Past players also like Diana with the imaging of the vascular structure in AML. Our collaborator, especially this work on nitosis, has been in close collaboration with Veni Papayopoulos that work on chronic infections, clinicians and patients for donating their cells to us. Thank you very much.
Thank you very much, Dominique. Great lecture. Very enjoyable. So I have a couple of curiosities because before giving chance to the attendees. One is kind of clinical question. Bleeding is one of the main causes of death in AML. And in the first part of the presentation, you showed that AML changes the permeability of the blood vessels. Yeah. This is the mechanism of the bleeding, or?
We haven't really investigated specifically that, but clearly, you know, I show you, we saw it's quite consistent. Whatever mutation the patient come up in our model, this is clear. So what we are trying to do, if we can get, I mean, we have clinical trials going on, but we have difficulty to recruit patients, is to confirm this leakiness in bone marrow of these patients by doing MRI. And we have the techniques that we have in-house. Doing that in mouse, it works very well. We can reproduce by MRI what we saw in imaging. We wanted to see whether this is real in patients, and we have only 3 patients in the trial so far, so it's going to be difficult. And then the question was, you know, can we use a NOS inhibitor or something else to treat?
That's the idea in this patient. Yeah. And the second was also a very particular question. When you mentioned the TET2 mutations, data I showed you, no? These are missense mutations, stop codon, frame shift.
Yeah, stop codon.
All these are stop codon mutations.
Yeah.
Okay. Yeah. Thank you very much. Questions here, Damiana?
Thank you, very nice talk. So I have 2 questions. One from the very beginning, from the MDS story, where you showed that the diseased MDS stem cells interact with the healthy stromal cells and then provoke this pathological niche. So my question is, this interaction has to be direct all the time, or you need first a direct interaction and later on then it gets a sort of cycle where you don't need
yeah, it's a good question. It's difficult. I mean, in vivo, they are in contact. I don't know whether it's direct contact all the time, for sure. We are doing also some imaging now of the scaffold. Because they are vessel also, so we wanted to see where they go specifically. So we don't know for sure. In vitro, we only have done this co-culture for a few days, so we also clearly see that if you do transwell wide away, it doesn't work. But as you say, I mean, the question will be, is it, you know, need to be constant or not? I don't know that for sure.
And then from the last part, and this really interesting evolutionary view of the leukemic cell and then the innate immune cells, which are the end, differentiated at the end. So my question with the dysfunctional neutrophils is whether you think what is more important, the homeostatic function of the neutrophils in the tissue, in the different tissues, or the immune functions of the neutrophils within the whole organism?
I mean,
kind of
question,
philosophical,
yeah,
but
I have no idea so far. I mean, clearly, neutrophil has tons of different actions. So we look at the most important one. So the infections is really something that our colleague actually is doing a lot. He has actually not invented, but have worked a lot on nitosis in the past. So he was really specialist on that. So we look very much into that because it's also clear that in prelocymic mutation, there's also some report to say that these people have more infection in general. So we wanted to see whether, nitosis and infection is linked. So we just wanted to associate potentially these and actually make the field know that it's not just the mutation in the HSC that are important, but the progeny could be as important and maybe more for certain pathology associated with that.
thanks.
Hi, thank you for the talk. It's wonderful. I have a quick question about the nitrocoxide signaling in the first part. So what is the role of the INOS, the inducible nitrocoxide synthase? Because I think that you mentioned the nitrocoxide 3,
Yeah,
but
INOS.
would your results suggest that if a MDS patient gets recurrent infection, for example, that get increased disruption of the epithelia and worse prognosis, something like that?
We haven't looked into MDS specifically. This work was mostly focused on AML. Now that we have a model to study and we can use this humanized mouse model with human endothelial progenitor cells, the idea is to do similar work to see, what are the effect of MDS on the vessel also. It could be happening, it could not be happening, I don't know. But the thing that we see was a clear signature, whether or not NOS could also be involved in other things and be also a stressor for inflammations. We don't know yet for sure, but at least what we know is that AML could induce directly that to the endothelial cells and without in context of any inflammation.
And maybe this is also to the second part when you use LPS, because LPS actually induces nitrocoxide, isn't it? So maybe that is a connection to that too.
Yeah, LPS is doing a lot of things, so we probably need to go more specific on other signals to understand what exactly is inducing what and what are the specificity of that. It was just that, a lot of people working on supposed infection put a lot of LPS ex vivo and mimic that. I think it's a bit easy, but it works for started something, and then we will focus more on the signaling that is important for doing that.
Thank you.
There is a question from the online attendees.
Yeah? Okay.
Thank you for interesting talk regarding the healthy individuals with CHIP, with clonal haematopoiesis. As far as know, they are described the presence of mutations. Has anyone described that these mutated precursor cells are different also in healthy precursor phenotypes morphologically? So in other words, do they resemble malignant cells by immunophenotype or cytology?
Yeah, I mean, so
It's done by Mata Crespo.
Yeah, I mean, that's what we're missing for the moment is the clinical link, because it's something we produce by CRISPR, this ingestion of our stem cells. So we are in discussion with the clinics at the present time, because clearly phenotypically it's almost easy to find the low granule neutrophil by forward and side scattered. And they have actually also markers that seems to be more enriched for these cells. So we are looking at these with the clinician that do FACS for MDS or AML, and also for potentially presence of prelocymic mutations. In our model, at least it works well. We could separate these, if we do this co-competition that I mentioned with or without LPS, without using HLA, now just looking at the neutrophil size and sorting the low granule versus the high granule and do a sequencing, the low granule is enriched in the TET2 mutant one. So we believe possibly clinically it could be useful, but we don't have the clinical data yet. So
thank you. I think there was somebody else in the audience before, here.
Great talk, Dominique. So I have a curiosity about the last part. So are neutrophils the only cells that are expanded and impaired in their function? I mean, among the myeloid cells?
no.
Okay. Yeah, I was interested on that. And maybe monocytes are also.
Exactly.
Yeah.
Yeah, we are, I mean, we needed to go somewhere. So we focus on neutrophil because there's nothing on neutrophil so far in clonal hematopoiesis. So wanted to have more novelty on that. But the monocyte, I mean, has been shown already in TET2 mouse model that monocyte also have some dysfunction. We are at the present time looking into this. There's clearly the differentially subset that I told you by the site of monocyte is one of the subset that we would like to investigate, and DC, which also nobody look at DC. I think we have also a colleague that is very specialist in DC, so we may end up going a little bit deeper also in DC.
Thank you.
Perfect. Thank you very much. In the interest of time, we should finish here.
Thank you very much.